All right, I think that ought to do. Okay. So, so where we were last time is we were getting a little deeper into this neural networks talk, and we were in the middle of talking about backpropagation. And so, just to get us sort of back on the same page, we you've probably heard of neural networks like the so-called convolutional neural network, or the CNN, which itself is very, very, very loosely nature-inspired or bio-inspired. So the basic idea behind the neural network, behind the convolutional neural network, is that you've got, say, an image, which maybe I'll draw this in kind of more 3D. So imagine that I've got an image that I'm looking at, and that image is broken up into tiny pixels. And so you would sort of want to say, well, how do I build a neural network that would analyze the pixels of this image? And so an approach that was inspired by looking at the human visual cortex, or actually, I guess we'd say the vertebrate brain. So in the visual cortex of the vertebrate brain, there are these regions, and they you can kind of think of them as layers. And so they go from V1 to V6. They don't quite go in a linear progression. So it's not like V1 goes to V2, which goes to V3, which goes to V4, which is V5, which goes to V6. There's a little bit more of branching and things going on there. But in general, you've got the ones with lower numbers are kind of closer to the image. The ones with higher numbers are kind of closer to the processing. And you'd also have this general pattern that V1, so each one of these things have these receptive fields. And we talked about the idea of a receptive field when we talked about radial basis function networks. And they basically have to do with how much of the image tickles the particular neurons in those layers. And so, V1, there are small receptive fields, which means that each neuron is, focused, I guess I might say, on a small region of an image. And as you go farther and farther back in the visual cortex, so as you get farther away from the image, then each neuron in each layer somehow becomes less refined. And so if you were to sort of say how much of the image could cause a change in that neuron, then the amount of image gets larger and larger and larger. And so initially, if you were to view the kind of, if you were to map this onto a neural network that you might build inside your computer, then it is as if you've taken this image and you've got a layer that within that layer, so each dot is a neuron in this single layer, and each neuron is sort of focused on this region of the image and only that region of the image, and the neuron next to it is focused on a region next to it, and so on and so forth. And so you cut the image up into small pieces, and then from there, then each of these pieces, so you'd have another layer of neurons, and let's say I'm just going to draw 4 neurons in another layer here, and then these 4 neurons, you can kind of view them as being connected to a subset of the neurons from the previous layer. So now I have a layer that is less dense, but it is effectively, this neuron is sort of sensitive to a larger region. And you could keep doing this further and further and further as you get farther down. And you can also imagine that, and I'll get to this question in a second, you can also imagine that these receptive fields can overlap, just like they kind of need to overlap in the radial basis function neural network. And so there might be, I've kind of drawn 4 neurons on this next layer, which kind of gives the impression from the picture I drew that these wouldn't overlap, but I could have made the groups, the clusters of the image from the first layer as they move to the second layer overlap. So you get neurons that are in the second layer are sort of have some bubble or some fuzzy region in between the areas of the image, and that can actually cause a change in both. And so these kind of overlapping receptive fields. So you have a small receptive field, and then over here you've got larger receptive fields. And then in general, we've got overlap between receptive fields within each layer. All right, and I see a question. Is it not also true for a regular artificial neural network that each hidden layer abstracts the features of the previous hidden layers into increasingly generalized concepts? Or is it something specific only to CNNs? So I'm going to talk a little bit about so-called regularization in neural networks. But basically, the idea here is that any deep neural network has got these hidden layers. The information is sort of flowing from one end to the next. But if you just have a general fully connected layers where one layer at so if I have a layer here that has as much complexity as the layer back here and is fully connected to the layer back here, it's true that as I'm moving from this point to this point, I'm effectively calculating a deterministic function. And so you can imagine as I go from layer to layer to layer, there is opportunity to sort of lose information. I can't ever gain information. And so that loss of information you can view as a coarsening of your focus. But in the CNN, there's actually kind of topological constraints on who can be connected to who. And that way you're enforcing you're making it much more deliberate. So if I just had a complicated function implemented at this layer and another complicated function implemented at this layer, and it just so happened that as I move from one function to the next, I end up, like you can imagine, like if a function maps every number to 1, then I've effectively gotten rid of a bunch of information. Or if a function maps all of the information to 0 and 1, I've basically turned a complex amount of information into 0 or 1. And so I have coarsened the world, but I didn't do it topologically, I kind of did it mathematically. So here we're doing it topologically in a convolutional neural network. And the thought was that it's kind of mimicking the way vision is processed in at least the vertebrate brain. And so it turns out that it's much more complicated to what's going on in the vertebrate brain, but this is the basic idea here. And when you draw it out like this, those of you who are working, say, signal processing or have done work with, image processing, then this might start looking familiar because it kind of looks like the mathematical operation of convolution. And convolution is just an operator that allows you to take a chunk of a very high resolution signal and aggregate over that chunk and then move that chunk to the next chunk and aggregate over that chunk and so on and so forth. And so as you're aggregating over a large chunk, the output of this aggregation that is moving kind of across the image actually smooths out the image. And so that smoothing of the image is what we can think of it in terms of filtering. And so in some ways, the convolutional network is filtering as you go down through here. So it is somehow taking noisy input here and extracting the important salient features back here. The same way you might take a noisy audio signal and run it through a filter and get rid of the noise. And as you get rid of the noise, then you can just hear a smooth signal coming out of it. That's hopefully what's happening here, is you will extract features which somehow are less noisy than the image that came in. And these are low resolution features as opposed to like high resolution features. So that's the basic idea behind this convolutional neural network. But the reason I'm bringing it up now is just to motivate this basic structure of the so-called multi-layer perceptron. So this idea that you can have layers connected to layers connected to other layers. So as you keep moving from layer to layer to layer, then you have this multi-layer. And technically, this term perceptron is really supposed to only be used for binary classification, but it has become more general in the case of to sort of talk about any multi-layer network that has this kind of structure, that has this feed-forward structure. So with some, over time, it has come to mean basically a feed-forward network, which has layer connected to layer, connected to layer, and so on, where there are no interlayer, there's no intra-layer connections, but you could potentially be fully connected between layers, but no connections within layers. And so the convolutional neural network fits within this structure. So if we can figure out how to train this structure, we can train a convolutional neural network. And so we were talking about last time that a method of training, or in this case, supervised learning, that we use is called backpropagation, which is just a clever realization that you can decompose gradient descent to make it much easier to train each layer independently, starting with the farthest back layer. So you can effectively train, I'll say, farthest layer, so farthest from input. And I'm going to say first, although that's not quite, I'm using this term very loosely, then move to successively, I guess I'll say, shallower layers. So we started talking about how that worked, and I won't go through the whole derivation again, but basically where we left off last time is we said that you've got this every single neuron. So recall that every single neuron has got some so this is, we can think of this as neuron I in layer K. And neuron I in layer K has an activation function. Oh, I'm sorry, I meant neuron J. Let me clean that up a little bit. And every neuron has its activation function. And the activation function takes as an input a weighted sum of all of the outputs of the layer before it. So we take a weight here, W, Lj, superscript K, and these are the outputs. And so here we've got Rk minus 1 is the number of outputs. Whoa. In layer K minus 1. And we have O. This is output L from layer K minus 1. And then this here is the weight applied to OL K minus 1 at this neuron. And so you bring in all these outputs from one layer to the next layer, you weight them all together, run them through the activation function, and so that's what's going on at every neuron. And so I am just, for simplicity, going to call this thing that is inside the sum here, or the sum itself that's inside the brackets. I am going to call that thing xjk, which again is just the sum of this. And then so now I just need to figure out how the heck am I going to train these weights. And so I picked a loss function. And so my loss function, for simplicity, I chose a nice quadratic least squares type loss function. And so I'm saying that that's E is going to be equal to, and I'm going to do 1/2 of, and I think I did Y hat minus Y squared, where this is a desired output. This is the actual output of neural network. And so we're basically evaluating this loss function for a given input. And so if we give a whole bunch of output or inputs and a whole bunch of outputs that each one of those inputs should correspond to, when we want to minimize this loss function. So we were projecting this data onto a space of weights for the neural networks, and we're choosing the best weights to approximate this input-output map for our data set. And so we could also rewrite that loss function as 1/2 the AJK, and then its argument minus Y squared. So I could just replace that Y hat with the activation function from that output layer. And so there we have it. So now I need to, I'm interested in the gradient of this thing. So I am going to just use good old fashioned calculus to try to reduce, to basically find a fixed point of the loss function. And so I am going to take the partial derivative of this loss function with respect to every weight. And that ends up giving me this beautiful decomposition by the chain rule, where it's just the partial derivative with respect to the nearest, or with respect to each activation function. And then each activation function with respect to its argument, and then each argument, which is just a weighted sum, with respect to the weight that I'm interested in. And this term here is just the derivative of the loss function. So you choose a loss function shape and you take the derivative of it and you can get that. So we know that automatically. So if we chose a sigmoidal loss function, then we just take the derivative of a sigmoid and we pop in the function for that. So that's known. This thing over here is just the output from the previous layer. And we are going to feed in, for every time we're evaluating this derivative, we're evaluating at a particular set of inputs. And so we can evaluate for the current weights the derivative of the activation function. Oh, I'm sorry, if I might have said loss function, I meant the derivative of, yeah, so this here, the derivative of the activation function, right? So this is the derivative of the loss function with respect to each weight, and that can be decomposed into a piece that includes a derivative of the activation function. Apologize for that. So this output here, given that, so if I start with a random set of weights, I can put those weights into the network and I can put data into the network, and then I can evaluate the output. And so that's solvable, I know that. So all that I'm left with then is this component right here. And there are two ways. So there's two possible expressions for that component, depending on if I am right next to the output or if I'm in the middle of the network. And so if I am at the output layer, so maybe I'll write output layer, then my derivative with respect to the weights is just going to be the output layer's activation function with a bunch of stuff I can evaluate minus the desired output. And so this is just the error. And that will be the beautiful part here in just a second. And then if I'm at an inner layer, so all other layers, maybe I'll write this in a different color, but I'll write all other layers. Then that partial derivative, oh, and yeah, I guess I screwed up. This is not with respect to the weights. I apologize for that. This is respect to the activation function. So this is supposed to be a JK, and as is this supposed to be a JK. So again, I'm just dealing with this portion right here. And so this here is going to be equal to, and I'm just taking the total derivative here. So this is going to be the sum of the partial derivatives with respect to later activation functions times the partial derivative of those later activation functions with respect to the partial derivative of the activation function I'm interested in. And if you think about it, this term right here is just equal to the weight W JLK plus 1, and so that's a constant that I know. And this term here is known from earlier calculation. In other words, if I bring the laser pointer up here, as long as if at the instant I want to calculate the gradients with respect to each weight, as long as I start at the weights, nearest the output layer, then I get this beautiful composition where the gradient with respect to the weights in the output layer are basically just going to be determined by the current error. So we bring the error in to the network. And then once I know all of those, basically these partial derivatives right here, I now have enough information to calculate the partial derivative with respect to the weights of the next layer back. So once I calculate this for the output layer, which involves calculating this for the output layer, I now have enough information to calculate this for the next layer back from the output layer. And then I will use this total derivative function here. And then I can keep doing that. Once I have one layer, I can keep pushing it back. And so I end up getting this nice pattern where I'm taking my current weights that I'm at in my gradient descent, and I take those weights and the inputs and I push them forward. So it's almost like I'm taking, I've got my, my current, maybe I'll write this in a different color. Maybe, yeah, okay. So if we got my current weights, I can then take the current weights plus the inputs, and I can push them forward through the network. And then I can take the error and then push it back through the network in order to calculate new weights. And then with those new weights, I can then recalculate the network, push it forward, and then take the error and then push it back, and so on. And so this is back propagation. And it just comes from the chain rule. Back propagation is the realization that for now, for a multi-layer perceptron, you've got this beautiful decomposition where the chain rule allows you to consider each layer independently, so long as you start from the back layer first and then move that back and forth. And so with that, you do that over and over again, and eventually you can converge on a set of weights for a set of data. And those weights can be viewed as regression coefficients, where the internal variables, the so-called latent variables in the hidden layer, are very strange combinations of inputs that somehow have a role in predicting the outputs. So it's basically just a big regression, nonlinear regression. Are we sort of ignoring bias right now? It seems like you need similar calculations for bias, or that's somewhere factored in here. So I am assuming that if we're referring to like the bias that we had in the single layer perceptron, I'm assuming that we've wrapped up bias terms as part of the input, that we have kind of a constant input and a bunch of bias terms. And so all that is sort of in the architecture of an individual neuron. How often do we need to run back propagation to get the weight to converge? Well, so that how often, how long until this gets convergence, it's very much similar to like the meta heuristics we've run. We don't know like how many cycles back and forth through here we're going to need in order to get convergence on these weights. We just kind of have to run it, and for certain optimization for certain data sets, it may converge very quickly, and for other data sets, it won't. You might stop early. So there are all sorts of ways to prevent so-called overfitting that actually involve stopping the back propagation before your weights settle out, and there may be some value to that. So, so I can't say ahead of time exactly how long it's going to take for this, but I can say that it's not unprecedented to just let the thing run until you reach some tolerance. I don't fully understand the partial derivative for all other layers of the error with respect to activation. I think you're referring to this big partial derivative down here. And so this is the total derivative And if you were to sort of go back and review from, from calculus, what the total derivative is that it's this basic idea that if if there are in if there are a number of different ways that the thing that you're trying to take the derivative of could vary, then you can take individual partial derivatives along each one of those directions, and then sum all of those things together. And so if I'm sitting at the top of a hill, the total derivative is basically the sum of all of the ways in which I can vary on the top of the hill if I kind of go in different directions, where here the different directions are these different activation functions that come from these different layers. So I am focused on a layer inside the network. And so in order for me to understand the partial derivative with respect to the layer I'm on, or the activation function of the layer I'm on, I am breaking that up into a multidimensional problem where I ask, how much does my loss function depend upon these other dimensions that are involved in the activation layer or the activation function that I'm working with? And so I know that my activation function gets wired to a bunch of different other activation functions in these layers that are closer to the air. And so I'm saying, how much then of all of the variance that, so it's kind of like, if I were to draw a picture here, if I basically have this loss function at the, I've got, let's say I'll call this my output layer, and I have this AJK that's back here, and I would like to know how much this loss function depends on this AJK. So in other words, if I were to make a delta AJK, I want to know what the corresponding delta loss function is. But this delta AJK can act on the loss function in different ways. And so in this intermediate layer, which is AJK plus 1, or AI K plus 1, I plus 1, K plus 1, and so on. So there's all of these activation functions that are at this intermediate layer in between the focal layer and the output function. And this delta here gets spread out among those. And so a little bit of it goes there, a little bit goes there, a little bit goes there, a little bit goes there. And then all of those get combined into the total air that I have at the actual output. And so what this total derivative is doing is it is basically saying, first, I want to know if I generate a variation up here, how much is that variation going to affect the output function? And then if I generate a variation here, how much is that variation going to affect the output function? And so on, dot, dot, dot. And once I know how much these little constituent variations add up, then I ask, okay, great, how much does this variation back here affect each one of these variations? And so now if I know how much the variation I care about is spread out among the intermediate variations, and then I also know how much the intermediate variations each affect the output, I combine those things together to finally get my answer of how much the little variation in the activation that I'm caring about affects the output. And so that's what the total derivative is doing. It's just splitting variation in different dimensions and then putting them all back together. And so that's like a visualization of the total derivative. And if you just go back to total derivative, and so the total derivative is, is the actual upright d, whereas the partial derivative is the squirrely d. So that's what we're trying to visualize here. And so you can think of these as partial derivatives, and each one of these is partial derivatives, and then you combine all the partial derivatives into the total derivative. Great. Any other questions about this general framework of backpropagation? And really gradient descent. Generally, when you're applying gradient descent, you're probably using backpropagation. All right, cool. So I mentioned that, oh, can loss be calculated with other? Yeah, of course. You could. I just, for simplicity, chose this least squares approach. And you could create other loss functions. And if you create other loss functions, then it may affect how you, so like, this layer back here. So at the output, this basically is the part that depends upon the loss function. And so if I had a differently structured loss function, then this thing would change here. But otherwise, this decomposition basically should apply for other loss functions as well. And so the basic idea of propagating the error back, the weights forward, the error back, the weights forward, should be able to be used for other loss functions too. It's just clean to use this one. And this is a common loss function to use anyway. Any other questions about backpropagation? Now, we have no idea if anything like this goes on in natural neural networks, of course. As I will talk about here, maybe towards the end of this lecture, there is good reason to believe that a great deal of neural networks are untrained in natural systems, and we'll see how that works. And so training, to me, I think that computer scientists and electrical engineers have kind of oversold the importance of training and have underappreciated randomness. And so given the way that I lectured about physics-inspired meta-heuristics earlier, you can kind of maybe anticipate that, that I think randomness might be important in neural networks as well. And I think people are starting to realize that in the literature. So we'll get to that. So anything else about backpropagation? No, it's a term a lot of people hear about, but I don't know if you necessarily see this. So I just want to make sure you see sort of, because I do think it's really nice. And this is something, if you were to do like high school outreach or something like that, like none of the math here is that bad. It's, it might blow the mind of like a 12th grader, but it would be something they could follow along with. And everybody hears about neural networks and things. And I think this is kind of cool because it really shows that like relatively basic math can allow you to do pretty sophisticated training on these complex neural networks. Okay. All right, so where do we go from here? So that allows us to think about these multi-layer perceptrons and how the heck to train them. But there are lots of other cool neural network structures that we might want to build. And so I talked about inspiration from the human, or from the vertebrate visual system, the visual cortex, from V1 to V6. And we can also think about inspiration from other neural architectures. So, so this is just more motivation. And in this case, I'm going to focus on the insect brain. And a lot of this generalizes to so-called pancrustacea, and even just arthropods and invertebrates in general. So there are aspects of what I'm saying that apply to a very wide range of taxonomic groups, but I'm sort of focusing on most of these insights come from thinking about the insect brain. So I bring this up because the insect brain is a very interesting structure. It's and I can actually show you the whole brain and actually map through every single portion of the brain, kind of a computational framework of how you get from a sensory system to a motor output. And you can, and I think some of you that already work with deep networks will actually see that a lot of the deep networks that you've been working with already look a lot like the insect brain. And it sort of suggests that there must be other ways that we could drill down into the insect brain to get insights in how we craft our own neural networks. So what does the insect brain look like and how are things wired up as far as we know? And if you were to look at a picture, if you were to Google a picture of the insect brain, and a lot of what I'm focusing on is in the brain of a bee, an ant, or a wasp. But this also applies to fruit flies and a bunch of other insects. But basically, an insect brain looks a little like this. You've got these two bulbs on the outside. And so this gets a little closer to stuff that we work in my laboratory, for example. And so, and these are optic lobes. So I'll just write OL for on one side and optic lobe on the other. And so these are like, kind of like right behind the eyes. They're pretty gigantic. And then next to the optic lobe, you have these little tiny things called lateral horns. So and they're lateral, they're on the sides. And if you were to zoom in on them, they kind of look like little horns. And then, and I'm going to talk about that a little bit, but not too much. And then there's these really cool structures that are very important called mushroom bodies. And they kind of look like inverted mushroom caps. And my pen has, all right, there we go. And so you don't really have to be drawing all this perfectly. I'm just kind of giving you an image of what's involved here. And these mushroom bodies are involved in sensory integration and higher order thinking and so on that we'll kind of get into. And then there's this thing in the middle called the central, so I'll write these are called mushroom bodies. And then this CC is called this central complex, which is used for navigation. And then there are these regions called the antennal lobe. And then there's a couple other things that I'm not going to go into, but I'll just mention that there's some other important parts that link all this stuff together. But that's basically your basic insect brain. Is you've got these sensory processing areas, the optic lobe and the antennal lobe. And then you've got these mushroom bodies, which, as we'll see, are involved in higher order processing. And then you've got this lateral horn, which is sort of involved in post processing. And the lateral horn ends up kind of making a lot of the decisions that go out into the motor system. And so, and as we'll see, that although it's not drawn away here, this I think actually takes the structure of a lot of the deep neural networks that are kind of used in state-of-the-art deep learning pipelines. And so what I mean by that is if a fly or a honeybee smells an odor, for example, so we've got the sensory system out here, and that sensory system has a bunch of odorants. So it has these, we'll say, receptors that can detect different odorants. And so you might smell some coffee. That coffee is a bouquet of a number of different odorants. And each odorant can be detected by these different receptors. And so these receptors, each one of them gets what we say projected. And so I guess I should define these terms. So there are two types of neurons you have in neuroscience. There are interneurons, and well, I guess there's three. There's motor neurons and there's sensory neurons, and those are the ones that are actually tied to the muscle or tied to the sensory system. And then there are these interneurons. And these are the things that connect neurons together. And of those interneurons, there are local neurons, and there are projection neurons. And the local neurons, they connect within each And then the term that people use is neuropil. And a neuropil is just a dense connection, I'll say a dense group of neural processes. And then the projection neurons, then they connect neuropils, or connect between neuropils and other structures. So, you know, if you feel a prick on your finger, then that thing gets projected to your spine, and that gets projected up to your brain. And so these long range connections are these so-called projection neurons, or PNs. And then, but then inside each one of these neuropils, which are like the antennal lobe or the mushroom body, then you get connections within, which are these local neurons. And so already we're seeing that, like in the feed forward, the multi-layer perceptron, you really only worry about projection neurons because you've got these layers, but these layers aren't really connected to each other. They're just connected. They're just if we think about it, we're just partitioning a graph up into it's like a bipartite graph would be two of these. We're just creating partitions of a graph where each partition only connects to another partition. And so there are only projection neurons between partitions if you think of each partition as a neuropil, but you could also have connections within those layers, and that's what these local neurons would be. And if once you did, then each layer would be a neuropil, and then the connections between them would be projection neurons. And so your sensory system, and maybe I will put this on a, I think I can maybe do this here. So in your sensory system, it basically is, it's out at the antenna, and it is smelling the physical world, and things are binding to receptors and innervating those receptors, and then suddenly those neurons project into a feature extractor known as the antennal lobe. And so there you get these projection neurons that connect to the antennal lobe. And so you can think of this as feature extraction. And so what do I mean by that? Well, it turns out that if you look at the antennal lobe, it's got all these little bundles, about, say, 50 of them. So there's about, around, say, 50 of these bundles that are called glomeruli. So a lot of this is just, the terms here are probably not going to matter too much to you, and I just want to, the fact that they're named shows that like they seem to matter at a discrete level. And the beautiful thing about these glomeruli is you can put a chemical into these things that detects when they're being innervated, and you can visually, under a microscope, watch them light up. So basically you have this chemical that each one of these glomeruli is tied to a receptor. So receptor terminates here. And so if you were to puff a whiff of coffee across the antenna of a bee and then have its head open, because you can do that, and are looking down at these antennal lobes, then suddenly a bunch of these glomeruli would light up using this special chemical, and many of them wouldn't. And so this basically becomes a pattern that is hopefully relatively specific to coffee or certain odorants inside coffee. And so you can do this in real time. There are people here at ASU that do this in ISTB1. They sit in a lab, and well, not right now, but before COVID, they were sitting in a lab and opening a bee up harness and puffing odors across it and watching these things light up. And so each one of these glomeruli were then projected to the mushroom body. And so this is, pardon the ugly drawing here. And they're projected to these little things around the edge of the mushroom body, of which there are about 200, and they're called Kenyon cells. And there's about, say, 200 of them. And so you go from 50 of these glomeruli, and you can think of them, if you're thinking about a neural network, you can think about these as like 50 neurons. And these neurons, you can think of them as encoding this odor that came through there, and some of the neurons turn on and some turn off. And so you've got this family of these like 50 that are like on and off, like ones and zeros, or you can think of as fractions between ones and zeros. And so now you've got that, and you can think of it like a bounding box of an image or whatever. You fed an image in and it gave you a bounding box out. You fed a complex odor into the sensory system and it gave you a code out. And these features then get expanded in representational space to these like 200 cells. And from individual to individual, the projections here actually vary. So each glomeruli might project into 4 or 5 different Kenyon cells, but those projections from one animal, from one honeybee, might be different than the projections in another honeybee. So internally, at the layer of the Kenyon cells, then these long range connections actually can vary from B to B to B. And then from these Kenyon cells, and I'll flip just to the next page here so I've got more room, but I'm just going to basically start, keep moving on this image here. So from these, so this is the same mushroom body I just drew on the previous page. So all this is just me just reproducing what was on the previous page. So these are the Kenyon cells. And there's about 200 of them. And we've got these projections from all of these different glomeruli. And then these Kenyon cells, and so I saw a question here. So the bees have their own idea of what coffee smells like. So that's an interesting question. This might be similar to the question of like that you and I both understand that there is a color orange. But what I view orange in my head to be might be different than what you view orange in your head to be, but it doesn't actually matter that our internal representations of orange differ, so long as we both agree that the same thing is orange. And so we can both communicate about it. So it's unclear to say if they have their own idea. They might have the same idea, but the idea might be just represented differently in the different brains. And then how do you know which Kenyon cells are triggered? You can actually put another chemical basically at this side of the Kenyon cell and see where it's wired. So just like in a house that you don't understand the wiring, there's ways in which you can go to an outlet and clamp something onto the outlet and then go down to the breaker box and see which breaker it's connected to. You can actually do that inside the brain. And so you can actually see, without tracing, like you could actually trace an entire neuron, but even without that, you can stain neurons and see where the stain goes, and you can map these things out. And so it's hard to see how the Kenyon cells are lighting up because they're kind of in a less convenient place, but you can do this staining, and that's how we kind of know about these things. And I see people have asking about Kenyon cells projection is different, but the projection on glomerula is the same for bees, is it? Yeah, so that's a good question. So the projection into the glomeruli, this we basically understand as kind of this is less diverse from B to B to B. It is not necessarily the same, but it is, it's sort of well known that there are certain, well, I mean, to think of it this way, there are certain device level characteristics at the sensory system in the antenna that are built for certain odorants. And each one of those devices at the antennal level, we know projects into these things here, and there's sort of less room for them to be rewired. But that's one of the beauties of having a deeper neural network, is you create a level of indirection where the connections here are these interneuron connections, have the ability to be more variable. And they say, who owns the Kenyon cells, bees or the, so I don't know necessarily what you mean by owns there, but I mean, they are an anatomical piece of the mushroom body, just like the fingers are the piece of the hand. When we talk about the mushroom body, the Kenyon cells are like fingers of the mushroom body. All right, so once you've got stuff in this mushroom body, in this expansion, then from there are additional kind of small regions, neuropils, that are bolted on to the mushroom body that I'm not going to get into too much detail here, but basically these Kenyon cells map to these other regions that are called alpha, beta, and gamma lobes. And so these are actually technically also still part of the mushroom body. And then those things map to the lateral horn, which we've talked about. And the lateral horn is also getting direct input from the antennal lobe. And then this lateral horn, then it finally goes out to motor neurons. And that actually is what makes the bee move. And it appears like this whole process also has feedback that feeds back into those antennal lobe local neurons. So if you are, so the beautiful thing about this antennal lobe representation over here is that it starts with a particular, so the antennal lobe down here with the glomeruli, it starts with a particular representation per odor that you give to the bee. But if you reinforce that odor with sugar, for example, then gradually other odors that are not reinforced but have components that are shared with the reinforced one gradually stop expressing the overlap with the odors that are reinforced. And so there's some cool feedback where you actually make the feature extractor better over time. So it's kind of like, if you've, I don't know, going back to the bounding box example, if you've got an image and you're extracting bounding boxes, but you find out that only circular bounding boxes are reinforced and square rear bounding boxes are never reinforced. And then it's almost like you go back to your feature extractor and you tell your feature extractor only to give you circular bounding boxes. And so it starts then only producing circular bounding boxes, or maybe it only finds things that look like circles. And so that kind of goes on. So there's some cool stuff there with feedback that I'm not getting into, but I just want to bring up there that although I've mentioned this as a kind of a feed forward network with local connections at each layer, there are connections in feedback that we're not currently exploiting and say, you know, in a reinforcement learning framework, like with convolutional neural networks that clearly are going on in real natural systems where you're getting tuning of the feature extractor over time. I cannot get the point why Kenyon cells in the mushroom bodies can't impact the bee. I don't know what you mean by they can't impact the bee, but it's well, let me get to what we think is going on. So from a computer science perspective, you might ask, you know, why would you go from, so you've got a sensory system with lots of pixels, so a very complex world, and you then go into feature extraction. So then there's feature extraction at the antennal lobe. And so now you've gone from very high dimensional to low dimensional. And then you go back out before you actually start doing the action maps. And so this is where you get kind of action mapping. Before you start mapping sensory input to actions on the output, you have a huge expansion in representation. So you get this large expansion. So these are your, you know, your roughly 50 antennal lobe or glomeruli. And now you go out and you've got roughly 200 of these Kenyon cells. And so you get this representational expansion. And it turns out that whenever you give a particular odor in, then that odor, let's say it is going to turn on a sparse set of Kenyon cells. And so that odor comes in and it might only turn on, like out of these 200, 3 Kenyon cells. And then you might give it a different odor. So, odor 2, and then that turns on another sparse set of Kenyon cells. And then you put together another odor, and then that flips on another sparse set of Kenyon cells. And I should, and I just want to emphasize this representational expansion, as far as we can tell, is random. And so, to those of you that have taken a data structures class or have a computer science background, does this look like anything? Does this look like something you've seen before, where you have inputs coming in and they end up getting a representation that is sparse in the number of representations you could possibly get, but kind of guaranteed to be different than the representations of other inputs? What does that kind of sound like? Does anybody have a thought or an idea on that? I see one answer. Yeah, hashing keeps coming up. That's right. So we think this is nature's version of hashing. And so we think that then this thing here gets mapped down into actions. And this is where we think a lot of the training or learning is going on. So we think there's learning going on here at the level of learning which features to extract. And we think that there is learning going on here, kind of trying to learn what representations to turn into actions, which actions to use. Do I turn left? Do I turn right? Those sorts of things. But this expansion here, we largely think is random, and we think the function of the randomness is to do exactly what hashing does, is to try to guarantee that if you give multiple odors in, that those odors get their own representation that is very far from any other representation, so that you can then confidently say, if something is represented by these 3 red lines, if I see all 3 of these red lines, and only if I see all 3 of those red lines, then I am going to do a particular action. And that prevents me from accidentally doing an action if I see an odor that might be similar, but doesn't have the exact same representation here. So it's this beautiful idea that randomness generates a hashing function. And if you think about it, we currently do a lot of this in our current deep neural networks. And so those of you who might study more of this, you see that, like, you've got these, let's say, an image, and then that image goes into a convolutional neural network, which produces an interesting set of features. And then you might have some of these fully connected layers that, and some other, processes that end up producing, some action or decision based on those inputs. So some sort of decoding or whatever here. And sometimes, you can play with these topologies here, where you could actually have representational expansions, just like the mushroom body does. But normally we think about training the entire network. So we, think about training the whole network. Or if we talk about transfer learning, we might have someone else train the convolution, the feature extractor. And after someone else trains the feature extractor and then publishes it, we might take their feature extractor and then plug it into our own neural network and then train it to do our function. But we never kind of leave some of the network alone. And we think that that's actually and there's actually evidence that the vertebrate brain does this as well, is that it actually creates representational variability or again, just representational expansion. And that expansion makes training easier downstream. And so it is a way to link feature extraction. So we also think that that nature adapts this over time. And we also think that this stuff adapts over time. But we think there's a lot of room for the stuff in the middle to maybe be effectively random. So there's a lot that we can, I think, learn from these neural networks, but I think it's promising to see that we actually have systems like the insect brain that already kind of look like networks we're building. And so that kind of suggests that we should be able to take from the insect brain and maybe just make minor modifications to how we're training our neural networks to actually get like artificial insect brains that might actually do better at the tasks that we currently hope that they'll do that we're training the whole network to do. So when people say ANNs, they're using loosely inspired. Well, so yeah, so this question of, I definitely can understand people that are very skeptical of saying that neural networks are nature inspired, because they are very loosely nature inspired, for sure. I mean, most of a neural network, of an artificial neural network, reflects more like a just human innovation than something that's being borrowed from nature. However, I don't want to totally just, I'd like to, I don't want to totally discredit that, because what I do think is interesting there is if we can find where there are commonalities, then we can start asking, why is this not a good model for nature? And if it's not a good model for nature, what could we make it, what could we alter to make it more of a model for nature? Like I'm saying, with this kind of randomness as hashing mechanism. And I think it takes people willing to kind of think of both types of neural networks under one umbrella. So to me, I guess that's the way I think of this, is there's some large umbrella of design. So this is my design umbrella. And we have, real neural networks over here and artificial neural networks over here. And it's true that these two might be very far apart. But if we learn to kind of view them under the same umbrella, then it is my hope that we will learn to bring them closer together. But I think it is only by recognizing that they're different, but they have the opportunity to be similar, that we can hope to close that gap. So long as we assume that the gap is already closed, we'll never close the gap. And I think that's a big mistake, is a lot of people over here assuming that it's already closed. And then also, I think by people assuming that the artificial neural networks are hopelessly non-nature inspired, then that also prevents us from closing the gap. So I think there's room for optimism in this design umbrella. And so my kind of goal, at least in my own research program, is to try to come up with frameworks on which we can bring these communities together. But that's kind of separate from the kind of syllabus in this class. But anyway, so where I'm going with this, any other questions about this basic structure before we get in back to the plain old artificial neural networks? But convolutional neural networks layers are not different at the architecture layer. Well, so I mean, there's, where could there be differences between natural systems and not? Well, for one, the device properties so can be different. So real neurons versus artificial. And from there, given that the device properties are different, then that also makes you wonder how much of an impact of the network topology is. So the big question is, if you have an artificial neuron, which really is just a generalized linear model, so a single neuron is just a GLM. So networks of GLMs, are they really, topologically speaking, do the networks have the same function if you swap out the GLMs and put spiking neurons? And so the question is, so how much functionality comes independently from the network? And I don't think we have a good answer for that. I think that when we look at, say, the insect brain, we can definitely, at a very, very high level, use the exact same reasoning we can when we look at a deep neural network. We can say, look, there's feature extraction and so on and so forth. But we have to be a little careful, because really what we're doing is projecting a model of how we do sensory processing and computation in our systems onto the bee. And for now, that model seems to work pretty well in kind of what we expect on things. But we might end up finding out that that actually we're all wrong in the architecture. And once we get closer to really understanding these differences related to spiking neural networks and so on, and neuromodulation, so your brain is constantly oozing out these different neuromodulators that change the weights of the networks at short time scales. And so once you kind of include all of these other feedbacks in there, then it may be that these topologies, we need a totally new way of thinking about it. But I think it's a good place to start. And then, the backpropagation thing, we don't think that backpropagation has any relationship to how things are trained. We don't know if things, my kind of point with this randomness is that I think individual neuropils are trained separately. So it's a little bit more like, training the feature extractor on ImageNet data and then applying it to images that aren't in ImageNet data, but you just don't want to go through the process of retraining your feature extractor. So I feel like in the brain, you're not constantly training the whole thing. You're sort of training little individual parts of it. And then those parts, work together, and then they get trained on how the other parts work. And so it's a very asynchronous process, but backpropagation is just it's a heuristic for us to make some progress in dealing with these giant, ugly neural networks. So with that, we're at 420. So where I'm going with this is to then back off these kind of complex examples from biology and say, all right, what more general structures can we build using our own human innovation? How do we train them with the same ease that we have with backpropagation in feed-forward networks? And that hopefully gives us a tool set for us to at least start talking about neural networks. And then I'll throw in some of the stuff about spiking neural networks and all the interesting things that come out of computation with spikes. So any other questions? I think I'm going to stop there on content today. All right. I don't have office hours today, but I do have them tomorrow at 4:30. So it'll be in my office hours link in case anybody needs that. And I'm working through your homework 1Bs, which I know you guys don't have grades for, and working through getting other assignments out and adjusting the assignment schedule relative to the days left in the semester and so on and so forth. So stay tuned. I don't have that much more to tell you at this instant, but I'll have more concrete data and adjustments, based on time left and so on and so forth. All right. So if there's nothing else, then I think I'm going to stop the recording. And okay.